like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the Yong Lin School of Medicine's Healthy Longevity webinar by National University of Singapore. Thanks for joining us. We have Rich Miller tonight, and I'm happy to introduce him in a couple minutes. I want to remind you to please use the Q&A function to ask questions, and we'll try to get as many of those to Rich as possible during the show. Today, before we start with Rich, we have Danielle Yo, who's an intern at Temasek Polytech, and she'll be talking about the effects of glycine on the aging cardiovascular system.
Thank you for the kind introduction. Good evening, everyone. As mentioned, I'm Danielle from the Center for Healthy Longevity as part of the preclinical team, and I'll be sharing the topic for my major project, which is the effects of glycine on aging cardiovascular system. The cardiovascular system is one of the 11 major systems that contribute to how the body functions. It consists of the blood vessels and the heart, and its main functions are to transport nutrients, hormones, gases, and waste products, maintain blood flow, and maintain body temperature. As a person ages, the cardiovascular system works at a less than optimal rate. The rate of decline of the cardiovascular system can be influenced by 3 different factors: physiological changes due to aging, environmental factors, or previous diseases or surgery that has deteriorated the body. These physiological changes include arterial wall stiffening, an increase in left ventricular wall size, which reduces the amount of blood that can flow through, cardiac fibrosis, and prolonged systolic contraction and diastolic relaxation. There are many subsets of cardiovascular diseases, also known as CVDs, such as coronary heart disease, thromboembolic diseases, which include embolisms and thrombosis, and myocardial infarctions, also known as heart attacks, and lastly, heart failure. Some causes of CVDs are hypertension, atherosclerosis, and hypercholesterolemia. There are 2 current ways known to reduce the risk of CVDs. One is lifestyle changes that we as individuals have the liberty to implement. Things such as regular exercise, cessation of smoking, reduced alcohol intake, and a low sugar and fat diet are all steps that can be taken in our daily lives to reduce the risk. Individuals that have a more serious risk of CVDs can be prescribed a number of treatments that help with the causes of CVDs. For example, antihypertensive therapies are prescribed to individuals that have hypertension. There are still many areas of research that can be explored to discover new prevention methods that are not specific to any physiological risk factors, such as hypertension, hyperlipidemia, or even diabetes. Since aging is a natural factor that does increase the risk of CVDs, the mechanisms relating to CVDs and aging can be explored to find out if there are any ways to reduce the risk as an individual ages. One such compound we are looking at is glycine. Glycine is a non-essential natural amino acid that acts as a precursor for many metabolites such as glutathione, creatine, heme, and purines. It is an important contributor to metabolism and nutrition due to its main function of protein synthesis. Glycine has also been found to have anti-inflammatory and antioxidative properties, which can improve overall health. We also naturally produce glycine through other compounds such as serine and choline, and it can be obtained through an individual's diet. Eating foods rich in protein can be a good source of glycine. Glycine supplements are also a good form of supplementation, since there are studies showing that glycine levels actually decreases as a person ages. A study on C. elegans supplemented with glycine showed significant lifespan improvement. Additionally, a study in 2019 showed that glycine supplementation has increased lifespan in mice. They fed mice with diets containing 8% glycine and found that compared to the controls, they lived significantly longer. However, they only focused their research on lifespan and did not touch on the health span of the mice. The health span of the mice is an important part to assess, as it ensures that the mice are not just living longer, but are living longer in better health. Clinical frailty studies were also not done, which can help to gauge the physical abilities and conditions that the mice might have. Thus, I'm working on investigating how glycine can affect inflammation, specifically in the aorta. To do this, I will be testing the aorta of aged C57 black 6 mice supplemented with different doses of glycine. We also plan on doing quantitative polymerase chain reaction testing for pro-inflammatory and anti-inflammatory markers. With the results, we will then compare the control samples with the glycine samples. We will also use immunohistochemistry to examine the structure of the aorta samples through staining with antibodies of interest. To supplement our findings, we also took a clinical frailty test of the mice at different time points, which can be used to further solidify our results. Thus, this project aims to study the effects of glycine supplementation on aging male and female wild-type mice and investigate its effects on the cardiovascular system to determine whether glycine has overall health span improving properties and to provide sufficient preclinical data for translation to human clinical trials. That brings me to the end of my presentation. Thank you for listening.
Thank you. And glycine, the paper that was cited was published by Rich in the NIA intervention testing program. So we'll hear more about that in a minute. They've identified a number of molecules that affect aging in mice. Before I say that, though, I want to remind you to continue to enjoy attending our Healthy Longevity webinar series from January 2023 onwards. Kindly re-register when prompted at the start of the next webinar. So remember to sign in early next week. The re-registration process should take about 2 minutes. We have to do this every calendar year. So please join us next week and re-register so you can hear the shows in January. I've known Rich Miller for a long time. He's a professor of pathology at the University of Michigan and directs the University of Michigan Glenn Center for the Biology of Aging. He's the head also for the University of Michigan component of the NIA intervention testing program. I already mentioned that a minute ago, but it's been an instrumental component of longevity research for a long time now. Rich did his postdoc work or his graduate work at Haverford. His PhD, he did his BA at Haverford, his MD and PhD at Yale, and postdoctoral work at Harvard and Sloan Kettering, and then joined the pathology department at BU in 1982 and moved in 1990 to University of Michigan. And his title of his talk is very simple and very important: Drugs That Slow Aging in Mice. Rich, thanks for joining us at 6 in the morning. It's you're a saint.
Well, it's an honor to be here. So I wanted to talk about two things today. Maybe the first third of my talk is going to bring everybody up to date on where the ITP has been going. And in the last 2/3 of my talk, I want to give some insight into a new, what I think is a more or less new concept, aging rate indicators. Try to convince you they're important and try to convince you that it's really very, very, very different from biomarkers of aging. So let's dive into the ITP. I'm not able to advance. There we are. So this is the published stuff. All the drugs that worked are shown over on the left, and if they worked in that sex, they're shown in red. Since all this is published, I won't go into it in any respect, except to note that the best drug in terms of percentage increase so far has been rapamycin. That will change, as you'll see in about 8 minutes. But in the females, rapamycin gives a 26% increase in median, in the males 23%. It was really puzzling to note that many of these drugs worked better in male mice than in female mice. Some of them didn't work in female mice at all. And I don't know why. We would really like to know that. We have some hints, but nothing definitive. So here are the updates. This is the stuff that is either unpublished or was just published a few months ago. The mice born in 2017, the headline is that if you combine two of those drugs, acarbose and rapamycin, you get our world record, a 29% increase in median longevity in the males. The females, it works just as well as rapamycin, but no better. This is also the first instance of when you combine two drugs and you get an effect that's better than either of them used alone. So that's good news. And other two drug combinations are probably worth checking out as well. Captopril gave a marginal, very small increase, about 5% in females, not in males. And we're going to retest it at a higher dose. Maybe it will work in both sexes. 5 other drugs listed there didn't work. This is what the rapamycin plus acarbose data set looks like. I started at 9 months of age. You get those nice red curves, and the control mice are the ones shown in the green circles. The 90th percentile age, which is our indirect index of maximum longevity, went up 19% in the males as well, which is impressive. Captopril is shown here. You can see in the males, it's not really impressive. In the females, it's also not really impressive, but it's statistically significant. So we're going to do some more work on Captopril. For 2018, we tried Phisonide, a drug that was alleged to remove senescent cells. It did not provide any health benefit at all, but it also didn't remove senescent cells. The next year, 2019, we had some winners, and I will show you the data on that in just a moment. The winners were Astaxanthin and Meclizine, both over-the-counter medications, and we found, for the first time ever, two drugs that hurt. Mycophenolic acid and 4-phenylbutyrate. They led to significant harm in female mice. So here's what the Astaxanthin drug looks like. In the females, it did not have an effect, but in the males, it led to a 12% increase in the median, highly significant. You can see that the curves come back together again in the very oldest mice. So there was not an increase in the lifespan, the maximum lifespan index. Here's Meclizine, which, like Astaxanthin, is available over the counter. It came from a screen that Gino Cortopassi did. He said that amongst the FDA-approved drugs and over-the-counter drugs, this was the best TOR inhibitor. So he thinks it's working as a TOR inhibitor. And as at the concentration we used, we got an 8% increase in the median lifespan. But again, for unknown reasons, saw this in male mice only. We're going to try Meclizine again at higher concentrations and see if we can get it to work more, and to see if we can get it to work in the females. Here are the drugs that are now being tested. This is the one that we'll get all the data we need in about 6 months. All of these drugs, none of this is secret stuff. All the stuff is posted, freely available on the ITP website. So if you have an idea for a drug you'd like to test and you want to know whether we're already testing it, just look up the NIA ITP website. It will list all the drugs for you. We're going to try canagliflozin again, which worked if you start early in life. We're going to see if it works starting in middle-aged as well. At this point, less than half the mice are dead, so I'm not allowed to tell you what's happening. But in a few months, when we have half the mice of both sexes dead, then I'll be allowed to present it in public. The oldest mice in the study is 31 months of age. Here's the list of drugs that we're testing with the mice born in 2021. As you can see, they are very diverse. One of them is an integrated stress response inhibitor. One of them works on pyruvate dehydrogenase. One of them increases cyclic AMP. You can read the list here as well. At this point, all I want to do is encourage you to think up some drugs you want us to test. There's an annual competition. Anybody can suggest a drug. The application is free-form and rather short. Examples of applications that worked are available for you on the ITP website. You can send trial versions of your application to me or to Randy or to David or Nadia for free feedback. It's more like a collaboration than a competition. But the only reason the program is done so well is that each year we get 5 or 10 really good ideas. And if you have drug ideas, you're allowed to submit as many as you want. We would love to talk to you about it and work with you on that. These drugs are in the pilot stage. The impressive work done by Brian and some of his colleagues on alpha-ketoglutarate, we agree it's impressive, although it was only done in an inbred mouse, so it's hard to know if it would work in a real mouse. We're using late start alpha-ketoglutarate in the 2022 mice, and in the 2023 mice, we're starting it early in life. The published data started rather late in life, and we're kind of hoping it will work even better if you start earlier. The other drugs being tested in the current yields crop are shown here. So the rest of the talk turns the page to a closely related topic that I'm excited about, and I'm hoping to get others interested in it as well. The problem to solve is it's not easy to guess which drugs will extend mouse lifespan. And so the challenge for this line of experimentation is to find reasonably quick ways to improve the frequency of good guesses. Right now, about 10 to 15% of the guesses submitted to the ITP by the best scientists in the world, about 10 to 15% of those actually extend mouse lifespan. So if we could get that hit rate to 20% or 30%, that would be really great. We would love to do that. So we took the following approach. We compared 9 different kinds of slow-aging mice as young adults and asked, what do these guys have in common physiologically? So here's the basic strategy. We had 4 kinds of mutants: the Ames dwarf, the Snell dwarf, PAPA knockout from Cheryl Conover's lab, and the growth hormone receptor knockout from John Kopchick's lab. We had 1 diet, the famous calorie restriction diet, and we had 4 drugs that we knew worked. Rapamycin works in both sexes. Acarbose works in both sexes, but it's much better for males than for females. And then we had 2 drugs, 17-alpha-estradiol and canagliflozin, that worked only in males. And the challenge is to find changes, physiological, endocrine, hormonal, someday metabolomic changes that are induced in young adults by all of these different kinds of anti-aging manipulation. Every one of these does tons and tons of stuff to mice, and a lot of it is non-overlapping. Our goal is to zero in on the stuff that is overlapping. All the work I'm going to show you was done by 2 talented researchers in my group, Gina Lee and Gonzalo Garcia. Most of it's published, but Gina has 2 papers in revision for Giroscience, which we're hoping they'll accept eventually. Let me show you some examples of what this looks like in practice. This is a molecule called GPLD1, and it's famous because when you exercise, you get a lot of it. It's made by the liver, and Gina found it's also made by fat and muscle and some other tissues made by the brain too. And it's known to go up with exercise in mice and people. So here on this panel, you'll see we take 2 of these long-lived mutants, the Snell dwarf and the growth hormone receptor knockout mice. DW stands for dwarf, GKO for growth hormone receptor knockout. And looking in the plasma, how much GPLD1 do they have? You can see that both mutations cause it to go up. The p-values are shown in the bottom of the ANOVA table there. It's 0.003 and 001. So the 2 mutant mice have lots of this in their plasma. Now, what happens if you look at the calorically restricted animals and the wild type animals? These are mice that are 12 months old. They've been given the drugs or the diet for 8 months since they were 4 months of age. The little asterisk sprinkled over that means significantly different from age-matched control mice that didn't get the drugs. The red triangles are females and the blue circles are the males. And the answer is, in the plasma for GPLD1, all 4 of the drugs plus the calorie restriction diet cause a significant increase. So this goes up in the mutants and the calorie restriction and the drugs. And what I'm not showing you here is that it also works for Ames dwarf. It also works for growth hormone receptor knockout mutants. It also works when you look at the liver protein. It also works when you look at the fat. All that's published or is soon to be published. Let's look at another example. It had been shown by another group that if you exercise, GPLD1 goes up, and if you genetically engineer a mouse with lots of GPLD1, they think better. Cognitive function goes up. And in fact, the brain biochemistry gets better. BDNF, which is brain-derived neurotrophic factor, that helps new neuronal plasticity and neuronal stress resistance. And double cortin, which is a sign of new neuron production, they go up in the brains of animals that have a lot of GPLD1. So Gina said, well, let's look at those in the brains of our slow aging mice. These are young adult mice. And you can see on the left panels here, double cortin in the Snell dwarf and the growth hormone receptor knockout mice goes up in both sexes to an equal extent. And in the right-hand panel, you can see the 4 drugs and the calorie restriction diet, and it goes up in all 5 of those non-genetic models of delayed aging in the hippocampus. So this slide shows double Cortin, but BDNF shows exactly the same thing. The Ames dwarf shows exactly the same thing, and the Pape knockout mice. So we're 9 for 9. All 9 models show this change, just as all 9 models show the change in GPLD1. Next, she looked at fat. This is uncoupling protein 1. It's been shown that if you elevate uncoupling protein 1 in a mouse, they live longer. So we wanted to ask the other question. If they're going to live longer, do they also have elevated UCP1? On the left, you can see the 2 of the genetic mutants, Gurko and Snell dwarf. You can see brown fat followed by inguinal fat, which is a white fat that's subcutaneous, and perigonadal fat, which is a kind of white fat that's inside your abdomen. And it doesn't matter. You have high UCP1 in brown fat, as well as in both kinds of white fat. And you can see on the right, with the exception of white fat in rapamycin-treated mice, that's our one exception to the rule. But except for that, caloric restriction and all 4 of the drugs increase UCP1 in brown fat and both kinds of white fat. It's not shown on the slide, but it's also true for the Ames dwarf and the Pape knockout mice. So UCP1 goes up. That's probably good for you. It helps you metabolically. Now, also in the fat, Gina looked at 2 kinds of macrophages. The M1 cells, marked by INOS, they're pro-inflammatory, and they're probably bad for you. The M2 macrophages, marked by ARG1, arginase 1, they're anti-inflammatory. They're probably good for you. So if you look here, the panel shown here is for the growth hormone receptor knockout mice. You can see that in both sexes, ARG1 goes up. That's a lot of anti-inflammatory macrophages, and INOS goes down. That's less of the pro-inflammatory macrophages. The same is true of inguinal fat, the same is true of perigonadal fat, the same is true of the Snell dwarf mice, and although I'm not showing it, the same is true of the Ames dwarf mice and the Pape knockout mice. You can also follow up on that M1 thing by looking at RNA for cytokines. And so what Gina did was looking at IL-6, TNF-alpha, and MCP1. I won't walk you through this. It says exactly the same thing. All 3 cytokines in both genetic mutants, and as it happens, all the drug mice go down, and that's presumably because they have fewer of the M1 macrophages. So here's the M1 and M2 macrophages in the drugs and the calorie restriction mice, and you can see the ARG1, that's the good macrophages, the M2, they go up in all 3 kinds of fat in all 5 anti-aging models. And INOS, the bad macrophages, go down significantly in all of these anti-aging models. Well, I won't walk you through it, but she found out that what's really happening here is not a direct effect on the fat. It's an indirect effect on the muscle, and the muscle sends something to the fat that causes these fat changes. That's published, so I won't go through it. But that was the motivation for her to look at a molecule in the plasma called Ireson. Others have shown that if you exercise, Ireson goes up in your plasma, and it's made from a muscle protein called FNDC5. So what's being shown here on the left is what you get if you look at Ireson. That's the top 2 panels, or and that's in the plasma. Or in the muscle, you look at FNDC5, that's the bottom 2 panels. For Snell dwarf and growth hormone receptor knockout mice, they go up. That is probably why all the good stuff is happening in the fat. I'll skip that. And we'll go on to the drugs and see our diet, and it's the same story, that you get an elevation of FNDC5 in the muscle and an elevation of Ireson in the plasma. Interestingly, at least for the muscle protein, there's a sex-specific effects for canagliflozin and 17-alpha estradiol. These are drugs that produce male lifespan extension only, and as it happens, the FNDC5 stuff only happens in the male mice treated with those drugs. So that's kind of cool. It might or might not be cause and effect. Next, we'll turn to Gonzalo Garcia's work. He's interested in a particular class of messenger RNAs, which are translated in an unusual process called cap-independent translation. I won't walk you through the details of that, but he was looking at 2 proteins shown in the upper left-hand panel here, NDRG1 and MGMT. He found that they go up in all the mutant mice. You can see the dwarfs on the top and the Gurkos on the bottom. But when he looked at the mRNAs, there's no change in the mRNA. So it was a non-transcriptional change. And it turns out that's because the mRNA level stays the same, but there's a lot more translation of it through a cap-independent message, cap-independent translation of this message. This is liver, but it's also true of muscle. It's also true of kidney. Then he looked at the drugs. So on the left here, you see acarbose, and acarbose does that. If you look at the first two bars, young is in black, old is in white. So there is an age-dependent decline in the production of these proteins that are translated through a cap-independent mechanism. Acarbose blocks that or reverses it and does so in both males and females. But if you turn your attention to the right, 17-alpha-estradiol opposes or blocks that age-associated loss of cap-independent translated proteins, but it does so only in the males and not in the females. 17-alpha-estradiol is a male-specific lifespan drug. And although I haven't shown it, it's in Gonzalez's publications. Rapa does work in both sexes. Canagliflozin works, but only in males. And Rapa works really fast. It works within 48 hours. I'll show you one more of Gonzalo's tricks. He also looked at a set of MAP kinases. If you don't know this already, I don't have the time to teach you about it, but I've put the little cartoon at the bottom there. This is a pathway called the ERK1 and 2 pathway, triggered by hormones and growth hormone receptors and insulin receptors. And there are 4 kinases in the pathway. And when you get out on the right-hand side, you're regulating translation by phosphorylation of an initiation factor called IF4E. And what you can see if you turn your attention to the Western blots is that there is an age-associated increase in this pathway. It's blocked by acarbose, whether you start acarbose early in light or late. It's blocked by rapamycin, whether you start the rapamycin early or late. But when you turn to a sex-specific drug like 17-alpha-estradiol, it's blocked in the males only. And that helps us to think that this particular MAP kinase pathway might be important in regulating translation in a way that promotes longevity. The other MAP kinase pathway is quite different. It's involved not in translation control, but in inflammation. Acute phase proteins come out the end of the pathway, and it's triggered by inflammatory mediators like the IL-6 receptor and the TNF receptor. In this case, rapamycin blocks it, acarbose blocks it, and again, 17-alpha-estradiol blocks it, but in this case, 17-alpha-estradiol blocks it in both sexes. So that suggests to us that this pathway may not be involved in lifespan extension because it does not parallel the sex specificity of the lifespan benefit. So to summarize this part of the talk, this is the current edition of our aging rate indicators. I'm hoping that a year from now, we'll have a lot more of them. These are our candidate lists, but the only things I want to point out today is that so far, we've seen changes in fat, brain, liver, plasma, muscle, and macrophages. We've seen them in 4 different kinds of genetically mutant long-lived mice, and we've shown them in calorically restricted mice and in 4 mice treated with any one of 4 drugs that extend lifespan and slow aging. And in some, but not all cases, the drugs are sex-specific. So here are the themes I'd like you to take away from my talk today. The first point I'm going to make, I'll show you a cartoon of this in just a moment, is that aging rate indicators are not biomarkers of aging. Every single paper we've ever submitted on this has gotten 2 positive reviews and 2 negative reviews. And the 2 negative reviews always say, oh yeah, biomarker of aging. We've got to get those people to stop reviewing my papers or to think about them more clearly, because aging rate indicators are not biomarkers of aging. Biomarkers are like a car's odometer. The further you drive the car year after year after year, the odometer keeps going up. ARIs are like the speedometer. It tells you how quickly the car is going or how fast the animal is aging. If you want to know how fast your car is going, do not look at the odometer. And if you want to know how old it is, how long it's been driven, do not look at the speedometer. You've made a mistake if you've done that. ARIs, aging rate indicators, can go up with age, go down or stay put. So here's an example. The thing at the upper left-hand corner is your famous imaginary biomarker. It changes with age. That's the blue line. That's the control group. And you give them a drug at 6 months of age. And now it continues to change. It just changes more slowly. That's what a biomarker of aging is expected to do. And critically, you can't tell if the drug has affected biomarkers of aging until they get old. They have to get old before you could see the biomarker effect. Looking at 6 months of age or 7 months of age won't get you anywhere. If you look at panel B, that's an aging rate indicator. This particular trait doesn't change with age at all. It doesn't go up, it doesn't go down, it just stays in place. And the drug is causing a change. In this cartoon, it's shown as an instantaneous change. Whether it's instantaneous or not is an important empirical question that we hope to look at soon. But there's a change, and the qualification that the drug is an aging rate indicator, alters aging rate indicators, is that there's a shift which is independent of the direction of the trait with age. Panel C, you see a trait that goes up with age. That's what an aging rate indicator drug would do. A drug that causes an aging rate indicator to change in panel D works for a trait that goes down with age. The direction of the change doesn't matter. And so you don't need old mice in order to accept my paper. You can tell whether a drug is working even on young mice. Second point I'd like you to think about is the changes in the proteins mediated by kappa-independent translation, though they were only a small part of my talk today. And also by something I didn't have time to squeeze in today, chaperone-mediated autophagy, which also is very important in aging. They'd be missed entirely by transcription studies. If you've got a huge transcriptome database from RNA-seq, congratulations, but you've missed about 70% of the changes in the proteome. And a lot of these changes in the proteome are regulated by either differential mRNA translation, for example, by kappa-independent translation, or potentially by differential degradation of the proteins. And the last of the general themes I want to bring out is that surprising number of these endpoints are not cell autonomous. I didn't show you the data, but it turns out that the brain BDNF and double cortin, they are modulated by molecules made by fat or liver, in this case, GPLD1. I didn't show you the data, but GPLD1 itself is actually activated by some mystery molecule made by some mystery tissue. It's not actually controlled by what's going on in the liver, something else, maybe in the brain, maybe in the vascular endothelium, gives a signal to the liver, which turns on GPLD1. Another example is that the fat and the fat-associated macrophages, that's again non-cell autonomous. It's caused by changes in muscle. So the key point for this audience, and I think for many audiences, are these are not modeled by drug screens in culture. They're not modeled by drug screens in worms, in flies, or in cell culture. If you've devoted a lot of attention to taking fibroblasts, giving them drugs, and seeing what happens, I think those are useful data sets, but none of these things would be picked up unless your culture happens to have like a brain and liver and fat in it. Those communications between tissues so far seem to be very important and have to be modeled in live animals. So the questions for future work to end up, I'd like to know how quickly these endpoints change after drug treatment. If we're going to use these to screen new candidate drugs and pick the ones that are going to work, I would like to know the answer to that in about 8 weeks. I would like to know, if you give drugs to mice and then wait 8 weeks, which of these aging rate indicators have already changed. We'd like to use that because we want to prioritize drugs that would deserve testing for anti-aging effects. So I was told I had 25 minutes. It's 24 minutes and 33 seconds. I would be delighted at this point to have questions from Brian and then from anyone else.
Thanks, Rich. Great talk. I've got a lot of questions here. Before I start, though, I just want to point out that scientific research is really Rich's hobby. His main job is to do nature photography. So you can see one of his photographs behind him. And you were telling me you've got another trip going in the near future. So where are you going?
Oh, I'm going with a group called Squiver to the Andes in Chile and Bolivia. If there's enough oxygen for the camera and me both to work, we'll get some nice pictures of salt flats and mountains. And it's actually the driest place on Earth. The Atacama Desert, which I believe you visited, has had no rainfall for 25 years. So I'll be bringing bottled water. Yeah,
there's 2% humidity when I was there. So yeah, bring a lot. Maybe we can start. You know, the ITP has been so useful to the aging field. I think it's brought a lot of people together around things that work to extend lifespan. How did it get started? What was the idea that led to the inception of ITP?
Huber Warner, who at that time was the director of the Division of Aging Biology, though it had a different name at that point. It was the Biology of Aging program. He got a group of 15 or 20 of us together in a workshop in Bethesda to say, hey, how should we screen drugs to see if any of them slow aging? The people who worked on dogs thought screening them in dogs would be a great idea. The people who worked on catfish, they thought maybe a catfish screening program would be terrific. Arlen Richardson and I went away and spent some time working on a program we liked, and we tried to sell it to the group the following morning, and Huber agreed with us. The key elements of the program were you had to have 3 sites so that if it worked on all 3 sites, you'd really believe it. And you had to have genetically heterogeneous animals. None of this black 6 nonsense where you have really no idea as to whether the drug would work in the next strain that you happen to pick off the shelf. So those were the 2 key elements. And the third element was that we wanted to have drugs suggested by anyone in the world as collaborators, and that we would therefore benefit from the community's collective wisdom and collective creative ideas. It took about 1.5 years for the NIA to approve the program. The first grants to myself at Michigan, Dave Harrison at the Jackson Labs, and Randy Strong in Texas were approved 19 years ago. We've had 20 years of funding with 1 year of exceptions. There was one study section that said, well, we're not so sure this is a great idea. So we had a 1-year hiccup. But with the exception of that, we've had 20 years of continuous funding. We're coming up for competitive renewal this coming year, and I'm hoping some of you will be on the study section if you like it. If you don't like it, then I'm hoping none of you will be on the study section.
So, one of the things from the early days that I thought was interesting is that the 3-site point was very useful, right? Because it turned out you would find subtle differences between the different mouse populations that were hard to predict in advance. Yeah. Yes. Surprising, but interesting. We do everything we can to regularize protocols. For instance, we all get the same drugs from the same supplier, made in the same kitchen at the same time, and shipped on 3 separate trucks. We all use the same source of bedding. We all make our mice at the same time from the same progenitor shipped to us from the Jackson laboratories. We do our very best. There's some things we can't control, like the water in Texas may taste different or have different contaminants than the water in Maine or the water in Ann Arbor. Temperature regulation is set by the people who own the animal colony. So there are differences, and these are probably really important differences of a couple of centigrade degrees between the warmest of the 3 colonies and the coldest of the 3 colonies. Consistently, year after year after year, with only 1 or 2 exceptions, the male mice at Michigan live 10 to 15% longer than the male mice controls at the other 2 sites. And we hate that, and we don't know why. It's not because of any differences in how good we are at Michigan at taking care of mice. Oh, you should take credit for it, Rich.
Well, it turns out there's data on that point. The female mice at all 3 sites always have the same, essentially superimposable survival curve. So we don't understand that. We also don't understand that in nearly every year, both males and females at Michigan are about 10% lighter in weight at their peak adult weight than the sex-matched control animals at the other 2 sites. So there's clearly some stuff that we wish we could control, but we don't know what it is, whether it's temperature, which is my current guess, or water contaminants, or smells, or subsonic sounds. We don't know, but it does give us 3 subtly different vivariums. So if a drug works at all 3 sites, that increases our confidence that it will work at your site too. And if it works at only 1 or 2 of the sites, then we publish it, of course. We think it's an important discovery, but we understand that there are unknown, mysterious, environment-specific variations that also influence the drug's ability to slow the aging process.
How often do you find things that work at all 3 sites? I mean, are the ones you indicated in red, they worked everywhere, or is that just a composite statistic?
It's hard to summarize it in a few sentences. Rapamycin always works at all 3 sites. Acarbose always works in all 3 sites. Once we move the dose of 17-alpha-estradiol up from after our original paper, we did another paper at a higher dose, a 3 times higher dose. Then it worked at all 3 sites. One reason it's difficult to answer the question is that if you are looking at the data from a single site, you have 1/3 as much data.
Yeah.
So your statistical power is much worse. If you have a drug that's giving a significant effect in the pooled data set, but it's an effect that's rather small, like glycine, for instance, an 8% or 6% or 10% effect, if you cut your number of mice in 3 by looking at one site only, it's much trickier to get that data set across the conventional statistical threshold. So we would never expect single site data to have nearly the same power as the pooled site data.
So you mentioned you've tested some combinations, and we've been trying to test combinations of drugs and natural products as well. And we're not very good at picking the right combinations. Do you feel like there's any rules or to this process, or are you have we learned anything about how to mix things together yet?
I think it's an important empirical question. Smart people like you and a lot of the members of the audience have different ideas about how to do it. We studied rapamycin plus metformin because we knew that rapamycin had a negative effect. It made animals more insulin resistant, which is bad for you. Metformin is supposed to fix that. And then we studied rapamycin plus acarbose because we knew that rapamycin was a TOR inhibitor and acarbose was working in mysterious ways, but probably ways that involved minimization of the peak daily glucose. That sounded kind of different, so we thought we would try those. In addition, acarbose worked so much better in males than in females, significant in both sexes, but much better in males. So we wanted to say, well, maybe the combination would also work much better in males. We agreed testing combinations is a great idea. We're now testing in the list of drugs that's on our website and that I ran past you all rather quickly. There's 3 or 4 more combinations which are rational. In some cases, one of our colleagues at another institution has said, hey, look, in my assay, if I combine these 2, I get an effect that neither does by itself. So maybe that effect would extend to lifespan and health span. Let's try it. That's a very persuasive argument if someone actually has some data. Another rational argument, we've had people say, I've got 5 different NRF2 inhibitors, and they are active on those systems, but they have different side effects. So if I gave you a blend of all 5 of them, maybe the good stuff would be complementary and the side effects would be minimized by the relatively low doses. That was a plausible rationale. And in fact, one of them did work, Protandim did work, though it was rather a very small effect. It was male specific and it didn't affect maximum lifespan.
Maybe one more question, then we can go to the aging rate indicators. Captopril, I think if I saw the data right, it affects the shape of the curve more than the maximum lifespan. So you get an effect on median, but not maximum. If you look across the things that affect lifespan, are there general differences in the shape of the curve, or something squaring the curve, or some things maybe affecting maximum lifespan more? I mean, is there a consistent, do you think there are differences is what I'm asking in the ways you can extend lifespan? And because you have a lot of data now.
I think it's an important empirical question, and that the data set is still too sparse to know for sure. It's very tempting to overinterpret the shape of the curve, because don't look at it as a line. Look at it as a broad, fuzzy cloud that is very swollen at the Middle Ages, and as you get to fewer and fewer live mice, it's extremely uncertain. So that if you repeat the same experiment 10 times in a row, the shape of the curve really is going to vary, even in rather large samples like ours. You can't look at one experiment and say, see, it's big at 700 days, but little at 900 days. There are several examples now of agents that appear to be working quite well at the median lifespan, and then they come back together again at the end. And we have examples of just the opposite, particularly if you start late in life, like at 16 or 20 months of age. There's no immediate effect. The median doesn't change at all, but the effect grows cumulatively, and by the 90th percentile, they're nicely spread. I think there's a hint as to what's going on here in a paper that just appeared in Science. The ITP people contributed to this by giving Johan Auerks and Rob Williams and Maroon Boo-Slayman and their colleagues 10,000 mouse tails from ITP mice of known age at death. And the first 4,000 were just published in the Science paper. One of the cool things in the paper that Maroon Boo-Slayman wrote showed that there are some genes which have a significant effect on lifespan, but only if you ignore the first half of the mice to die. This is something that David Harrison and David Burke and I published about 10 years ago, but on much smaller numbers of mice, 600 rather than 4,000. So I believe their data more than I believe our own data. But that's a hint that there may well be physiological processes as indicated by these segregating genes that have an effect only on the things that get bad after approximately the median age. So it might be that there are drugs that address those issues only. That would be rather interesting. You can also tell by looking at what happens when you compare drugs early in life versus late in life. Rapamycin, early in life and late in life, works just as well. No better, no worse. Acarbose, however, works great if you start early. If you started in middle age, it still works, but about half as well. So that's a hint that it may have differential effects on young and old animals.
The ARIs are really interesting. I think you go down the list. The macrophage data is striking, and maybe it's not completely surprising, but it's striking, I think, and it fits a lot of people's models on aging of anti-inflammatory effects. UCP1 similarly, has been seen to go up in a lot of interesting ways, and so that's intriguing. A lot of other stuff. One of the questions is, it's tempting to try to put that puzzle together and maybe fill in some gaps by looking at other things. Do you think there's a sort of a systemic network here that you're defining that you can put together a puzzle ultimately?
That is entirely the goal. There's a lot of reasonably obvious, straightforward things that I would do if I had, 5 more postdocs. Right now, Jin is doing it all with her bare hands and a couple of talented undergraduate students, but there's an awful lot more to do. Some of the most obvious low-lying fruit. How about the macrophages that aren't in the fat? I'd like to know about macrophages in, say, the bone marrow and the spleen and the lymph node. How about the glial cells, which are sort of like macrophages? Are they also modified by these anti-aging drugs and diets and genes? You would also like to know what is upstream. There's clearly something that is triggered by the diet, all 4 drugs and all 4 mutants, that is lying above this entire network of aging rate indicators. And what is lying above and controlling them is sort of, in a metaphoric sense, the holy grail. We would love to know which of these things, either things we've already measured or things that are not within the battery we've already measured, is leading to this coordinated pattern of change. Connecting these, as you've noticed, nearly all of the aging rate indicators have the obvious potential to contribute to resistance or susceptibility to specific diseases, UCP1 and obesity-related diseases and strokes and atherosclerosis and diabetes, FNDC5 and Ireson to all the fat stuff, but also potentially to muscle stuff, all the cognitive function that goes on in the brain that is modified by liver or fat-produced GPLD1. You can begin to construct downstream stories connecting these to diseases. There's one actual data fact I was planning to mention and forgot to, so I'll squeeze it in here. It was really cool to discover that GPLD1 is a cap-independent translated protein. That links Gonzalo's lovely stuff on the cap-independent translated CIT proteins going up with all of these 9 different kinds of models to Jin's stuff on the GPLD1 protein and its effects on cognition in the brain. I imagine we will stumble across or dig up many other examples of interesting proteins that are controlled by cap-independent translation. PGC1 alpha, a famous protein, is among that group, for instance, as is TFAM, a controller of mitochondrial biogenesis.
So, you talk about speedometers and odometers, but I don't know if you watch Ferris Bueller, Rich. It's possible in some cars to roll the odometer back. Do you think that can happen in aging?
Yeah, I think it's an interesting speculation. I am unaware of, I'm rather on the skeptical end of things because I don't have a product to sell, but I'm rather skeptical about the notion that something can rejuvenate anyone. That is, I don't think you can turn me into someone as young and vibrant and healthy as, say, you and Juan Pedro de Magalhães. I think getting me back into that physiological state is not going to happen. You can clearly improve the health of old people by getting them to do a lot of push-ups or by having them lose a bit of weight or eat a lot of broccoli and that sort of stuff. There are ways of getting specific functions systems to work better. I'm unaware of any experimental evidence suggesting you can actually reverse the aging process and dial it all back to a younger age. But in your hypothetical situation, if you could do that, it would be interesting to know what happened to these aging rate indicators, wouldn't it?
Yeah, but sure. I think, that's one of the fundamental questions in basic biology now, and something we're trying to go back and rethink about is, can you really reverse the process or not? And I think everybody's has their opinions right now. We don't have evidence yet, and we don't know the ultimate answer. We may have to think about it completely differently in the end of the day to find some approach to do that. So lots of still basic science to do in aging, and lots of big questions to answer. I want to bring
up the most surprising things that ever came out of the ITP, surprising to me, was that rapamycin works even in middle age and old age, 20 months of age. I would have bet a lot of money against that, but I would have lost the bet. I was really surprised and thrilled to see that it worked even in middle age. Now, whether it is, we know that at least in young mice, some of these aging rate indicators flip to the favorable position within 48 hours of rapamycin.
Yeah.
We haven't looked at old mice. That's a really important experiment to do, and to look at it with other drugs too.
Yeah, sounds good. Let me bring the audience in. Donglu's one of the grad students in my lab who's doing some great work on ovarian aging, and today she's going to be moderator. So thanks, Donglu.
Thank you, Brian, and thanks very much, Prof. Miller, for the great sharing. I'm going to start with some general questions from the about the ITP studies first. Our audience are interested to know if Healthspan is assessed in the ITP studies. If so, what are actually measured?
Right. We deliberately chose not to measure Healthspan in our first run-through. That is, our first run-through is one where we have a drug we've never tested before. We want to know if it increases lifespan in the mice. If we had added Healthspan indices to that primary screen, it would have been expensive, and we would not have been able to test as many drugs. Our strategy is, if we get a drug that actually increases lifespan, we publish that, of course. We're hoping that the brain experts will then look at, does this drug do good things for the brain? The muscle experts, the bone experts, the special senses. We then do, usually almost always, another study, a second follow-up study, which we'll call a stage 2 or phase 2 study, in which we make a lot of mice treated with that drug. And those tissues are available to anyone. Anybody who wants, we kill the mice at 22 months of age. We bank 11 tissues. It's publicly available on our website. Anybody who wants to can say, I want to find out if my thing is changed by this drug. Please send me a lot of muscle or a lot of heart or a lot of brain. We'll do that. The Healthspan studies, that is looking at things that don't hurt you, but can be measured without cheaply on 500 mice at 3 different sites, that's proven very challenging. That is, we have found some things like grip strength and rotor rod behavior and body temperature that are changed with age in both sexes at all 3 sites. But when we did our first attempts to measure these on mice that were part of a longevity cohort, the numbers at all 3 sites were radically different, and it was impossible, despite our best efforts, to mix the data from all 3 sites into a statistically powerful sample. Maybe it can be done, but it's really much, much harder than one might imagine. So we save those for the follow-up studies. Pardon?
That is really a practical consideration behind this, and we really look forward to any like Healthspan measurements in the future.
Well, they're all public. We publish them when we do them in the phase 2 studies. Those are published. For instance, for acarbose and 17-alpha estradiol, my colleague Mike Garrett, who used to be in my lab and now is in New Zealand, Mike looked at grip strength, he looked at body temperature, he looked at rotor rod, and he showed, for instance, for 17-alpha estradiol, it was great for male mice and not for female mice in improving all of these fitness measures. We have a new colleague at Michigan, a woman named Catherine Kazarowski, who's an expert on cognition. And one of the first things that she will be doing, I hope, in the next 5 years, if the ITP were by good fortune to be funded, will be to do some cognitive function tests on the mice treated with ITP drugs. Our hope is that they'll improve cognition, but we'll find out.
Just now you mentioned something about the experiments were done in males, and many of our audience are actually interested to know what are the hints behind the gender differences in the drug effects.
Sure, me too. There is no good general answer. As far as I know, it is a major unsolved problem. We have a few hints, but they might not be right. For instance, if you give mice rapamycin, the blood levels are much higher in the females than in the males. This could contribute to the apparent higher benefit in females than in males. If you give 17-alpha estradiol to mice of both sexes, it's converted by males into a different class of steroids, the estriols, and the females don't do that. So it's possible, speculating here, it's possible that the estriols are the good stuff and that the drug works in males because only the males have the steroid dehydrogenases that metabolize 17-alpha estradiol into an estriol. One can also make up more convoluted stories of there are major sex-specific differences. For instance, in the famous xenobiotic metabolizing enzymes, the liver turns out, dozens, 50, 60, 70 of these, and they detoxify things that you've eaten, like, say, drugs, and they are usually 5-10-fold higher in females, both humans and mice, than in males. Some exceptions, but that's the general rule. So it's quite plausible that the xenobiotic metabolizing enzymes may preferentially conjugate and lead to excretion of some drugs that are not treated in this way by males. All this is speculation, and real evidence on this point would be important to look at. Puzzlingly, the QTL studies, I mentioned the work of Rob Williams and Maroon Bousleiman and Johan Auwerks and their colleagues. Puzzlingly, some of the QTLs segregating genes that modulate lifespan in these mice are sex-specific. They give you insight into life expectancies in one sex, but not the other. So there may well be sex-specific pathways which are being revealed by these genetic studies, as well as by the pharmacological studies that we've been talking about.
Thank you for sharing this speculation. Now I would like to move into some more questions about specific drugs and that may set some fire. Okay, so there is an audience that is asking about Phisonide. So Prof. James Kirkland's talk at the previous NUS webinar sounds much convincing of Phisonide as an effective senolytic. What is your thought over the stark difference between the ongoing efforts to develop Synolytics and the results obtained by ITP?
Jim Kirkland was the one who recommended that we test Phisonide. We used the dose he told us to use. He told us to try it 2 different ways, continuous and also cycles, in which they got 3 days on, 11 days off, 3 days on, 11 days off. We did it his way. It had no lifespan effect. We were sad. We gave him tissues that we had taken at 22 months of age. We coded them so he and his colleagues would not know which ones came from treated mice and which ones from untreated mice. He then measured beta-galactosidase positive cells, and he also measured at the RNA level 6 things that he thought were characteristic of senescent cells. There was no change by Phisonide on any of these markers. We then sent tissues to another laboratory. I don't know that I'm allowed to name them at this point, but they repeated these studies and found no change. So we conclude that Phisonide at the dose Jim told us to use and with the schedule that he told us to use does not remove senescent cells from mice. As to how Jim and his colleagues find stunning results with them is, to me, a bit of a puzzle.
Rich, just out of curiosity, we did a small experiment with Phisonide and saw some changes in weight in the animals. Did you notice any weight differences with it?
I'd have to go back to my data. Send me an email and I'll look it up and send you the graphics. I don't remember. My own personal opinion. Now those are the facts. Moving away from the facts, I don't think senescent cells have anything to do with aging. I think the notion that senescent cells cause aging is a collective delusion based largely on hype and commercial interests. We can go into that the next time I'm invited here, if I ever am invited here.
We'll make the title Hype and Commercial Interest.
Yeah. I don't think senescent cells, I mean, they're critically important in cancer and wound healing and a lot of stuff. I think senescent cells are really great. I just don't think they are the cause of aging or in fact are related to aging in any important sense.
Yeah, I think their contribution,
it wouldn't work.
there's not much evidence that it's a big, if it is, it's a relatively small contribution. I think that the issue that bugs me is nobody's been able to ask the question of how much of the inflammation that happens with aging is contributed by senescent cells and how much of it is coming from other changes during the aging process. And
I think that's a first step towards asking a critical question. But if I were in charge of thinking about that, I would say before you can discuss it in an intelligent way, you have to define senescent cells.
yeah, that's also a problem.
Each lab that works with senescent cells defines it operationally in a different way. And sometimes their operational definitions change from paper to paper or from day to day.
Or from cell type to cell type.
Or from cell type to cell type, or from circumstance to circumstance. I think if you want to ask, are senescent cells responsible for the inflammatory change that's bad for you when you get old? Your first step is to say, okay, what do you mean by a senescent cell? I heard a really nice publication. It was in the Gordon Conference, so it's not something that one can discuss in great detail. But there's a guy who said, here's the definition of a senescent cell. I'm going to look at the RNA-seq data from many tissues of mice of many ages, and I'm going to count the number of senescent cells in each tissue as the mice got older. And his result was there are no senescent cells at any age in any tissue of mice. I think that's maybe a stretch, but it depends on what your definition of senescent cells are. P16 cells do go up in multiple tissues. Whether any cell with P16 in it is senescent, that's the sort of community definition. But Jim Kirkland, who's smart and sophisticated, says, well, no, there are an awful lot of senescent cells that don't have P16, and there are a lot of P16 positive cells that don't meet my current definition of senescent cells. I think that kind of more nuanced thinking is an important step. I believe once one has taken 10 important steps in that direction, people will be able to give up on the notion that senescent cells are important in aging.
All right, Dongley, I'll shut up and let you go on.
Yeah, we need to rush because we have a bunch of questions.
We probably have about 2 more, so pick your favorites.
Yeah, maybe the audience favorite, because there is a highly voted question about adenoviral gene therapy, because there has been recently more and more reports of lifespan and healthspan extension of adenoviral gene therapy. Have the NIA-ITP considered this option? What are your thoughts?
No, the answer is no. There are several reasons. I think gene therapy is a growth, deserves to be a growth topic. The issues you will want to have, of course, are, hey, what genes? What genes do you think are going to extend lifespan? Oh, and what tissues? There's no good answer as far as I know to that. If someone comes up with a good idea about a gene in a tissue that deserves to be moved up or down by viruses, in principle, that's worth doing. The ITP won't do it because you'd have to have a separate control group treated with null viruses or something. And we have to use the same control group for all of our drugs. Also, we can't use anything that needs to be injected, every week or every month. We could, in principle, evaluate an injectable if it was done just once, but only drugs in food or water in practice are testable in our protocols.
One more, just a moving forward kind of question. Currently, how close do you think we are in finding practical and realistic therapeutic treatments in the ITP studies that can be used to improve both healthspan and lifespan in humans?
Oh, the answer is negative 15 years. We published in 2009 a practical treatment. Now, if the question is, how long is it going to take to get these approved by the FDA? That's a political question, has to do with who can lobby them and whom you can bribe, and drug companies, and whether they'll believe dog data. It's a much more complicated. I think the sneaky answer is going to be screening over-the-counter medications. The rules, at least in the United States, are FDA must keep its hands off over-the-counter meds. Once we have a good repertoire of over-the-counter meds that work great, that the FDA is not permitted to mess with, I think people will start to take them, even though the evidence for human efficacy will, at that point, still be absent.
Yeah, I agree with that. I think that's going to be one clear avenue. Thanks a lot, Rich, for doing this. We'll have you back, and the next show will be all about hype. Have a good trip to Chile. I want to remind people to use the chat function and the panelists and all attendees option to leave comments and feedback. Look for news from our center on our School of Medicine as well on the in credits. Our center is constantly looking for members who are driven and passionate about longevity research to join our team. Check out the various career opportunities in the link provided in the chat box or scan the QR code at the end of the webinar. Last September, you may know, we launched our NUS Center for Healthy Longevity Clinic at Alexandra Hospital with a full-day hybrid conference featuring key leaders in the field of longevity science. If you couldn't make that, you're in luck because there's a live recording up now and you can find it in the link on the chat box or you can scan the QR code. I also want to point out the contribution of Leo Drago and Antonio Hui. Without their great sponsorship, we would have a hard time making this webinar work. They've been great benefactors for the center, and we're very grateful for them helping out in that regard. Our next and last episode of the year is Sergio Ruiz, who's co-founder and COO of Turn Biotechnologies, and I want to single him out because we had a late cancellation and he agreed to jump in on very short notice. His topic will be mRNA technologies and rejuvenation. I'll be there again next week, and at the end of the show, I just want to leave you with a 112-year-old birthday celebration from someone in the UK. Thanks a lot for joining us.
Happy birthday to you. Happy birthday to you.
To her friends, and she's collected many over the years, she is simply amazing Grace. Full of fun and happy to answer a question that applies to no other person in the country. What does it feel like to be 112 years old?
I don't feel any different. 65.
You feel 65?
Yeah.
That's amazing.
I don't feel old.
Born in Liverpool in 1906, Grace has known 26 prime ministers. She puts her longevity down to the nightly tot of whiskey she still enjoys.
My little drop of whiskey. I don't drink anything else. I don't drink in the day.
Marrying her late husband Leonard was the highlight of her life, but she says each decade has been special.
I've loved them all. Everyone.
And it looks to me like you're still loving them.
I hope so. I'm not going yet, I hope.
Grace hopes there'll be more parties in the years to come and that being positive, avoiding stress will help that happen.
Don't worry. I don't worry.
Do you think that's part of the secret?
Definitely. If you worry, it gets you down. I never, ever worry.
Whatever her secret, Grace's friends won't be at all surprised if they're all here again this time next year.
Isn't that nice?
Paul Davis, ITV News, Worcestershire.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.